Hello, everyone. You asked for it, so here is the review for Star Biology. I will be covering few important topics, and I've also posted some videos for you to visit. So, the first thing is biology is the study of life, right? So, what are the characteristics of life? What do you look for in an organism to categorize it as living? So the first and foremost thing is that it should be made up of cell. It could be one cell or it could be multicellular. Then it needs to reproduce. Reproduction can be of two types. It could be asexual, where they do not need a partner, and a lot of times they create exact replicas of themselves. And sexual is what gives genetic variation, and the offsprings are different from the parents. So reproduction can be asexual or sexual. Metabolism, all organisms, they require energy. They break down food to carry on their metabolic activities. So that's another criteria for being living. The next one is homeostasis, maintenance of internal environmental conditions, and this is done by positive and negative feedback loop. Majority of the feedback loops in all systems are negative feedback loop. Positive is a rare one because it is something that is a runaway loop and does not come back to its original state. Then it's hereditary, that is DNA, the genetic material that codes for is a common genetic code and is there in all organisms and is said to be a universal code. Okay, and the next one is responding to stimuli, whether it's plants or animals, all organisms they respond to stimuli, whether it's biotic or abiotic. If you think about plants, they respond to like thigmotropism, light tropism, gravitropism, so it's all response to stimuli. Then any organism that is born is doesn't stay in the same state, right? There has to be growth as well as development, and that growth and development happens through mitosis. We did that in class, so I hope you remember that. And then evolutionary adaptation, they all change and evolve based on environmental uh, stress, and it's all about survival of the fittest. There are more. These are not just that these are the only characteristics that are of life, but there are many more. Like interdependence is another one, where all organisms are interdependent on each other for their own survival. Well, the next one is what are living things made up of? They are made up of four main biomolecules. It's carbohydrates, proteins, lipids, and nucleic acids. You do not need to know the structure of these four biomolecules, but the monomers are important. So for carbohydrate, the monomer is monosaccharide. When two monosaccharides are joined together, they are called disaccharides. And when you have multiple, a very complex uh, carbohydrate is called a polysaccharide. And the classic example would be starch. Okay. Proteins, they are very versatile. Their building block or monomer is an amino acid. There are 20 known amino acids, and we do talk a lot about protein synthesis. I hope you remember that. You can reflect back on that. Uh, all the hormones that you have or the enzymes that you have, all of them are protein, and without that life would be very slow and would be difficult for all organisms. Lipids, it could be fatty acid, and they provide energy, and they can be of two types. It could be saturated or unsaturated. Okay, and saturated, usually they are found in animals and they are solid at room temperature, whereas unsaturated ones they are liquid at room temperature, and they usually come from plant based, like all the oils that you see around you, like olive oil or corn oil, or I don't know, you can tell me what other oils are there, vegetable oil, all these oils that are plant based, they are liquid at room temperature. Okay, and the last most important biomolecule is nucleic acid. It is made up of nucleotide that has three parts, five carbon sugar, a nitrogen base, and a phosphate group. Now, these nitrogen bases, when we do DNA structure, we do talk a lot about it. Is cytosine, guanine, adenine, and thymine. And then they usually store the genetic material, and they are of two types, DNA and RNA. There is a slight difference between DNA and RNA. DNA is a double it's a double layered helix, whereas RNA is a single strand. DNA's sugar is called deoxyribose, whereas RNA is called ribose. And the most basic last difference would be that DNA has a nitrogen base called thymine, whereas in RNA it's called uracil. Another thing is that carbohydrate gives you instant energy immediately, whereas lipids, they are stored up energy that you can use for long-term energy. Well, the cell theory, there are three parts to it. All cells are the basic unit of life. They come from pre-existing cells, and all organisms are composed of cells. So these three parts make up the cell theory. Cells come from pre-existing cells was the last part to be added to the cell theory. There are multiple organelles that perform very specific function for the cells. A few basic ones are like chloroplast. It performs photosynthesis and is found um, in plant cell. Mitochondria is found in all cells and they perform cellular respiration. They are the site for cellular respiration that produces ATP. Ribosomes are involved in protein synthesis. Vacuoles are a bit different in plant cell and animal cell. Plant cells, they have one large vacuole in the center, whereas animal cells, they have multiple small vacuoles. Okay. Nucleus, it contains the DNA and it participates in many activities of the cell. And the nucleus is the site of ribosomes formation. So now at the bottom, if you see that it talks about like, if you see it's the difference between plant versus animal. Plant cells, they have a cell wall that is more made up of cellulose. Okay. Animals do not have cell wall. Mm, Plants, they have chloroplasts where you have photosynthesis taking place. And animals, they do not have chloroplasts. And then I already talked about the large vacuole in plant cells. Okay, cells can be either prokaryotic or eukaryotic. Just remember that bacteria are the only organisms that are prokaryotic. They are said to be very ancient. They are found in almost all climatic conditions, whether it's like very hot, very cold, methane laden, they're everywhere. And when we talk about bacteria, they can be both beneficial for different organisms. They may be like leading a symbiotic relationship with other organisms, or they could be a pathogen. So, what makes a prokaryote a simple organism is that they do have a DNA which is circular, but it's not bound by a membrane. It's just like suspended in the cytoplasm. They do have, all of them do not have a flagella, but usually you will see a diagram that shows flagella. It's like a tail-like structure that helps the organisms move, and a lot of cilia. They also have pili or pilus. Pili is plural, that helps them to adhere or to stick to surfaces. And it also helps in conjugation, where two bacteria, they exchange genetic material. Bacteria, they reproduce by binary fission, a very simple division of bacteria. So they actually create an exact replica of themselves all the time. And since they are small and they're simple, they really like duplicate at a very fast pace. Okay. Then eukaryotic cells, they are said to have actually evolved from prokaryotic cells. It is called endosymbiotic theory, where you have it is said that chloroplast and mitochondria were actually a cyanobacteria and an aerobic bacteria that entered another bacteria, stayed there, started helping in the functioning, and that gave rise to eukaryotic cells. Okay, so it's a theory that scientists think could have happened. All right. So eukaryotic cells, they have their own DNA that is surrounded by a membrane, which is called the nucleus. And then you do have, like, if you look at them, almost all the other kingdoms like this Protista, Plantae, Animalia, and fungi, they all are made up of eukaryotic cells. They all have different types of organelles that all perform different specific functions, and they help in maintaining homeostasis. Okay. All right, the next thing we are going to discuss is cell membrane. So think about it. Anything that is made up of cell will have this membrane, okay? Another name is plasma membrane. It surrounds every cell. It is made up of phospholipid bilayer. So there are two layers, okay? Lipids, they are hydrophobic. They are non-polar, so they don't like water. Whereas the phosphate is hydrophilic, so it faces the outside of the cell. They also have a lot of other proteins that are embedded, a lot of carbohydrates that are embedded, surface proteins are there. All of these help in the movement of materials. So your cell membrane is semi-permeable, or it is selectively permeable. It selects certain organisms to come in and to leave, and it helps in the maintenance of homeostasis. When we talk about cell transport, it could be either active transport or passive transport. Now, passive transport happens naturally. It's the movement of materials from high concentration to low concentration, and no energy is required. So, diffusion of gases, of liquids, all of that is movement from high to low. Osmosis is a type of diffusion, but it's unique. It is when, when water moves from high concentration to low concentration, but across a membrane. Okay, so that helps in maintenance of homeostasis, and it is a passive transport. A lot of times when materials are not able to move in automatically, they are helped by either carrier protein, or they are helped by that is called facilitated diffusion. The next one is active transport, that is moving against the gradient, where you have particles or materials or chemicals or molecules moving against the gradient, that is from low concentration to high concentration. And this extra push requires energy, that is called ATP, adenosine triphosphate. Now, on your exam, you could maybe see some like where they give you a beaker with a cell inside and give you different concentrations. So just pay attention to the water molecule molecule concentration. So wherever it's high, it'll move from high to low, and then you can talk about it. If water moves into the cell, it is said to be hypotonic, and if the water leaves the cell, then it is hypertonic. And if water molecules movement is equal and is happening naturally, it is said to be isotonic. You can look up a video for hypertonic, hypertonic, and isotonic in order to understand it better. Okay, any I hope you all remember that enzymes are a type of protein, but they're very unique. To be honest, I don't think living organisms could live a healthy life without enzymes. They are everywhere. They are said to be biocatalyst, and they speed up a reaction in a cell. Okay, so they bind to the substrate. So if you see over here, this green is the substrate. It goes and it attaches to the enzyme at the active site. They all have specific shape. It's more like lock and key. It goes and it joins the active site, the substrate joins the enzyme. The enzyme changes shape, and then the product is formed. So once the product is formed, it's new from the substrate, and then it just gets released into the cell to be used by the cell. And there are a few unique features about enzymes. They are substrate specific because of the unique shape. So it's like a specific shape will only fit into it, okay? The next thing is that they can be reused again and again and again, all right? And the last thing is that they are very delicate, means they are delicate because it's called they get denatured very easily. It could be extremes of temperature, it could be extremes of pH, or it could also be the concentration level. All that has an impact on the functioning of the enzymes. And if it gets denatured means the shape changes. The shape over here that you see at the active site, that gets D-shaped, and because of that, the substrate is not able to fit in. And when it's not able to fit in, what happens is that the product is not formed, and when the product is not formed, then the cell just suffers and is not able to get the important molecules it needs for its normal functioning. Okay, these two cellular processes are important. You will be asked about the reactants, the products, and where it takes place. So just try to remember that photosynthesis takes place in the chloroplast of the cell, and only plants have this unique organelle. Okay, and mitochondria is present in all cells. So cell respiration itself means all cells respire, they make ATP, and they use that molecule to perform different functions in their cell. So let's look at this. This is the formula for photosynthesis. You have carbon dioxide plus water that reacts in the presence of sunlight to give you glucose and oxygen. Okay, so the products are glucose and oxygen. Some people also call it organic molecule. Some say that it's sugar, but any name is good. Okay, and then if you look at aerobic respiration's formula, you will see that the same glucose is broken down in the presence of oxygen to give you carbon dioxide, water, and ATP in the form or energy. So you get about 38 ATP for every glucose broken down. Now, that's just an approximate number. Both these processes are very complex. We just write this in formula, and at this stage, you need to know the reactants and the products. So make sure that you recognize these and you're able uh, to find the right answer. So, cell respiration takes place in mitochondria, releases energy, and is called aerobic respiration. That means it needs oxygen. There is another one where oxygen is depleted. At that point, they start doing anaerobic respiration. Okay, this anaerobic respiration is not very efficient. It does happen in your body, and it does happen in all organisms, and it happens in plants as well. So let's look at the difference between aerobic and anaerobic respiration. Aerobic requires oxygen, makes a lot of ATP, and is very efficient, and it produces products like carbon dioxide and water, and it happens in the mitochondria. Okay, whereas anaerobic respiration, it does not use oxygen. It is also called fermentation, and it makes a very small amount of ATP. So in plants it is called alcoholic fermentation. It produces alcohol, whereas in animals it produces lactic acid, and it happens on this in the cytoplasm. So these are key differences between aerobic and anaerobic respiration. Okay, there are two types of reproduction, asexual and sexual. I think I did mention it in the first few slides. First slide, I think. Asexual is simple. It doesn't require a partner. There's no genetic variation. And the cell divides very quickly. And usually bacteria and unicellular organisms, they perform asexual reproduction. They are like exact replica of their parent. Whereas nature requires genetic variation, and sexual reproduction is actually favored by nature. It involves meiosis, it requires fertilization. You have male and female gametes, you have two parents involved, and usually there is like alternation of generation. So what's the basic difference? Your cell divides in two different ways. Okay, these are both cell divisions. Mitosis happens for growth and repair. Okay, so when an organism is growing and it needs a regular repair, so the mitosis happens, and they create exact same cells as themselves with the same chromosome number, and they're identical. Like liver cell will produce liver cell, skin cell will produce skin cell, And there are four main phases: prophase, metaphase, anaphase, and telophase. You can remember this as PMAT, and it is the M phase of the cell cycle. Whereas meiosis, it makes a cell divides twice and it results with half the chromosome number, and they create sperm and egg. So in females, it is called oogenesis, and in males, it is called spermatogenesis. As a result of spermatogenesis, you get four sperms that are haploid and have half the number of chromosomes. And the result of oogenesis is okay. Let's talk about deoxyribose ribose nucleic acid, that is DNA, and it is one of the nucleic acids. Okay, it is a polymer of nucleotide. So the monomer is a nucleotide that is made up of the 5 carbon sugar over here, the phosphate group, and the nitrogen base. And this nitrogen base can be cytosine, guanine, adenine, and thymine. And if you notice the hydrogen bonds, there are 3 hydrogen bonds between cytosine and guanine, and 2 hydrogen bonds between adenine and thymine. Whereas in RNA, you'll not find this thymine, you'll find uracil, URACIL, uracil. Now, if you notice the side of the ladder is made up of this 5 carbon sugar and phosphate, and the rung of the ladder is made up of the base pairing. Okay, so a lot of scientists actually contributed to the understanding of the DNA structure. It was very complex, and the same DNA structure is found in all living organisms. It is said to be a double helix, and it is twisted. Now, DNA replication, if you all remember, it happens in the S phase of the cell cycle, and it is said to synthesize DNA. So DNA copies itself, it gets ready for division. Therefore, there are multiple steps to it. So if you see this one, this is the parent molecule over there, and it is where adenine is paired with thymine and cytosine with guanine. So the first step is that the DNA helicase comes and breaks the hydrogen bonds between the nitrogen base pairs. Okay, and once that happens, you have another enzyme, that is DNA polymerase. It comes and adds the base pairs, the complementary base pairs, to the 200 DNA strands that are acting as a template. And this results in two DNA strands. And if you notice, one is still the old one. See the dark color? One is old and the other one is a new one. So it is said to be semi-conserved. And they do this to maintain the DNA base pairing, and it is said to be semi-conserved, like I said, and it happens in the interphase. Now, interphase is made up of three parts: G1, S, and G2. Okay, and this happens in the S phase, and two enzymes are involved: DNA helicase and DNA polymerase. And DNA polymerase also does another job of moving back and forth, and seeing that no mutation has taken place. If there any errors are there, it's going to rectify it. Okay. All right. This is protein synthesis is most probably the most complex topic in biology, I would say. So try to understand this. They give you a lot of questions where they ask you to use the codon chart that is based on mRNA. Okay. So protein synthesis has multiple names. It's also called gene synthesis, gene expression, and central dogma. So if you know that DNA, if you remember this, it's kind of makes it simple for you. DNA makes mRNA, and mRNA comes with a message. This messenger mRNA comes with a message, and it gets translated into a protein. Now, DNA never leaves the nucleus, okay? So this whole process of DNA making mRNA is called transcription, and it happens within the nucleus, all right? And mRNA, after it is formed, it needs help of transfer RNA, that is called tRNA, that brings the amino acid, and it also needs the help of ribosomal RNA, this blob that you're seeing over here, that's the ribosomal RNA. So it needs the help of other molecules. Therefore, it leaves the nucleus and comes into the cytoplasm. Okay, so translation happens in the cytoplasm on the ribosome. All right. So let's talk about transcription first, where the mRNA is made. DNA unfolds and RNA polymerase comes and breaks the hydrogen bonds once again, and the two strands separate. One acts as a template, the other is a non-template. It just stays there, it does nothing, okay? So the template one adds the complementary base pairs, RNA polymerase adds the complementary base pairs for RNA. Now here the trick is that cytosine will combine with guanine and adenine will pair up with uracil. Remember, RNA has uracil, it doesn't have thymine, so it'll combine with uracil. Once the mRNA strand has been made, it drops off and RNA winds back again to its original shape. And mRNA then leaves and comes into the cytoplasm. Here, if you notice, they ask you a lot of questions. They'll give you like different pictures and they'll ask you to identify it. Okay, so this structure that you see over here is tRNA. It has anticodons at its tip that helps us to attach to the codons. Codons are triplets. They are three nitrogen bases and they are called codons, and they are made of three triplets, okay? And at the tip they have the amino acid. So all this amino acid, it forms an amino acid chain, also called polypeptide chain, that forms a certain type of protein. Now over here, when we are talking about it, this whole process starts with the start codon, that is AUG. And AUG is the amino acid is methionine, okay? But there are three stop codons. It's UGA, UAA, and UAG. So they will be providing you with a codon chart. You do not memorize it, but you need to know how to use it, okay? And it always corresponds to mRNA. Remember that, never to DNA. Now over here, again, it's the same thing, DNA forming RNA, RNA to protein, and these are the different steps given to you. I've already explained it to you, so I'm going to move on to the next slide. Like I was saying in the previous slide, that they ask you to identify the different parts, so I would suggest that you stop the video here and then try to identify all the different parts, okay? So you have 1 through 10, okay? So this one, what is this? So it's a double strand. This is a single strand. This is the nuclear membrane, okay? This is the nuclear membrane. So this process is happening in the nucleus. And then this strand leaves the nucleus and comes to the cytoplasm. This blob is where the translation takes place, okay? And look at this. These are the anticodons, all right? So what is this structure? What are these structures? And then what is this chain called? So I gave you a lot of hint to help you in identifying these molecules. So here you go. These are your answers. First one is DNA. Number 2 is mRNA. 3 is nucleus. 4 is cytoplasm. Number 5 is ribosome. 6 is codon. 7 is anticodon. 8 is tRNA. 9 is amino acid. And 10 is protein or the polypeptide chain. Okay? Okay, now let's see. How do you read this codon chart? It'll be given to you, like I said, you will not have to like memorize it, but be sure that is mRNA. There should be no thymine in it, okay? So you don't have to memorize this. So what amino acid is coded for by the DNA? So it's DNA. Can you use this codon chart for DNA? You can't, right? So you need to convert this DNA into RNA first. So adenine will combine with uracil. Thymine with adenine, adenine with uracil, guanine with cytosine, adenine with uracil again, and guanine with cytosine. So once you have that, there you go. You need to look at the codon chart now. So UAU. So this is the first base, second base, and third base. So UAU over here is tyrosine. Okay. So it'll be here. Then CUC. So this is CUC. So CUC is leucine. So the next amino acid is going to be leucine. So this is how you read the chart, and just be mindful that it relates to mRNA. Okay. Now, genetics. Well, Mendel is said to be the father of genetics, and he was the one who actually solved the mystery of recessive traits. People were confused that suddenly something would skip a generation and a trait would appear. So, a few key terms are important for you to understand. That is dominant, recessive, phenotype, genotype, homozygous, heterozygous. Okay, so you need to know that each chromosome has two alleles, okay, one from mom and one from dad. And depending upon being dominant or recessive, it deciphers the traits, okay? So over here, homozygous is expressed whether it is, sorry, a dominant trait is expressed either as homozygous or heterozygous, whereas the recessive trait will be expressed only in homozygous state. Okay, so here it's telling you that uppercase R, that is homozygously dominant, heterozygously dominant would look dominant, and recessive would be only when it is homozygously recessive. So over here they have given you a monohybrid cross. Okay, it's just one trait where they are talking about the color of the flower. This is heterozygously dominant, and the female is also heterozygously dominant. This results in three purple and one white flower. So there was no white over here, but it gets expressed when two recessive alleles are paired. So the chances of having a white flower is 1/4, and purple is 3/4. Now, if we look at the phenotype, it's going to look different because the genetic makeup is different. Both of them are purple flowers, but if you look at the genetic makeup, it's different, right? This is homozygously dominant, this is heterozygously dominant. Therefore, the phenotypic ratio is going to be 1 is to 2 is to 1. All right? Okay, Gregor Mendel, he worked with peas and he got just completely dominant or completely recessive, and he was able to solve a lot of mysteries. But you also need to know other non-Mendelian traits. Okay, so just be mindful that there are non-Mendelian traits like codominance, incomplete dominance, sex-linked genes. Okay, and at the bottom over here is your dihybrid cross. There is a shortcut for dihybrid cross too. I have put a link in here. You can watch the videos. I have actually put two links over there, and it makes it easy for you to come to a conclusion. Okay, so make sure that you watch those videos and you learn the shortcut method. It's kind of easy. It's not that difficult. You just need to know the genotype of the male and the female, come up with the gametes, and then put it as Punnett square. Okay. The sex linked is very unique. It's usually the female is the carrier. It's on the X-linked chromosome, and usually the traits are common in males, like color blindness or hemophilia, whereas female is usually the carrier. The female can have it though, but the thing is that the female needs to have it on both the X's in order to get the disease. Okay. Codominance is where both of them are shown or both of them are present. Like if you have a black hen and a white hen, the product white rooster, in that case, the product or the offspring could be speckled with white and black specks on it. Okay, so blood type is an example of codominance and of multiple allele as well. Okay. pedigree. You need to know how to use this chart, okay, how to read the chart. They'll not ask you to make it, but just make sure that you know how to read it. The square over here is male, the circle is female. If it is completely shaded, that means that male or that female has that particular trait, okay? If you have two lines, horizontal lines mean they are married, whereas the vertical line means that that's the number of children they have had, okay? And then try to stop this video and say, can you identify the genotype of individuals 4, 7, and 12? So 4, 7, and 12, okay? The genotype, the genetic makeup, not the phenotype, okay? Here you go. 4 is heterozygous. Number 7 is also heterozygous. And number 12 is homozygous and is impacted by the recessive condition. Karyotyping. Karyotyping is pretty much like a chart that is created where it's like the chromosomes are mapped, and it's a very useful tool. You know, the last one you see over here actually helps in identifying whether it's a male or a female. If it's a female, that'll be both X's and both will be of the same size. Okay. If it's a male, one, the X chromosome is longer than the Y chromosome. Okay. And can you identify the defect in this karyotype? Is the gene mapping? If you notice, all of them are pairs. Okay. But this one over here, the 21st one, is a triplet. It's three together. In this case, it's an example of Down syndrome, and the person has an extra chromosome because maybe during meiosis, the chromosomes did not separate and they came together. So this is called trisomy. There's something called monosomy also. Okay. So a lot of diseases can be identified by karyotyping. There you go. Genetic technology. Not very long ago, we didn't know much about genes, but after the discovery of genes and DNA and nitrogen bases, we have changed a lot of things around us. We have manipulated genes, we have come up with genetically modified organisms. We have the capacity as scientists to uh, make new organisms. Okay, so we can actually combine two different kinds because genetic makeup is same, right? It's a universal code that is similar, made up of the four nitrogen bases: cytosine, adenine, guanine, and thymine. So you can pretty much combine it with any organism. So we have used bacteria to make different types of medications for humans, like insulin. You incorporate the desired gene uh, part into the bacteria, and you let it multiply. And once it is multiplied multiple times, because it multiplies at a very fast pace, then what you do is that you isolate it and then use it as a medication. This is gel electrophoresis. It's a way of mapping, again, like genes, and then you use it in a lab setup, and then you can match the DNA of different individuals. This can be used for multiple things. It can be used for, I think, like for identifying parents, or maybe if there is a crime scene, they have discovered some sort of like evidence, and they can match it with different people who may be suspects. So this gel electrophoresis is used a lot and is called DNA fingerprinting. This next topic is evolution. So it's a theory. A lot of people, uh, when I was a student, we learned it very differently that monkeys changed into humans, and Charles Darwin was, this is called the father of evolution, and he came up with this theory. People made a lot of fun of him because they were not able to understand what he was trying to convey. Then now it is called natural selection, that organisms change over a period of time, and only those who are the fittest, they survive and they reproduce. So there are a lot of like evidences for evolution. And so if you notice, this fossil is one of the evidences. Then also looking at different types of proteins, also looking at homologous structures. So all of these tell us that there is a common ancestors that are there, and we all have evolved based on different the environmental uh, conditions. So let's look at this one. Um, the first one is fossils. Over time, you can compare different types of fossils and see that um, the similarities or what organisms looked like in the past. Homologous structures is also a very strong one. If you look at the forelimb of whether it's a bat, a bird, whale, penguin, monkeys, you'll notice that we have the same bones. We have the same bones. You'll be mighty surprised that the structure is the same. It may be fused in penguins as flippers, or in bats as long phalanges where they have membrane and it helps them fly. But we have the same bones. So that those are called homologous structures. That tells us that there is a common ancestor. The opposite of homologous is analogous. They do not provide us with any common ancestry. Like a bee, it can fly, right? And a bat can fly, and a bird can fly. But those structures, all of them wings help them to fly, but there is no like homology, or there's no like common ancestry over there. Vestigial structures, they tell us of some like function any organ had and it has lost it over a period of time. They say appendix is said to be a vestigial organ. At some point, tonsil was said to be a vestigial organ. If you look at the whale, they have a suspended pelvic bone in their body. That is a vestigial organ. So it just says that maybe organisms have changed over time and those organs have lost their function, but they're still there to tell us that they at some point they had some sort of function. Then you have embryology. Lot of embryos while they're growing in their early stages, they look the same. You look like a chicken at some point. Did you know that? So it's like interesting to see that. Then biochemistry, the amino acid sequencing. The least the difference, the more closely organisms are related. If there's more difference, then they are more farther apart. Like you there is a difference of one amino acid between humans and chimpanzees. Okay. The next one is adaptive radiation. Adaptive radiation is where one species it diverges into multiple species, and a classic example is finches. When Charles Darwin he went to the Galapagos Island, he collected and he was able to bring back a lot of specimens where he talked about finches changing because of the resources available. Some finches would eat insects, some would eat like seeds on the forest floor, some would eat seeds from the fruits. So they all had different beaks, and then they all had diverged from a common ancestor. Then we have another theory of Miller and Urey. They simulated Earth's environment and they said that they had created organic compounds like amino acids. And this theory was a forerunner for a very long time. It is called Urey and Miller's theory. Okay. Then we have this endosymbiotic theory. I think I mentioned it in the beginning, there where it is said that prokaryotes gave rise to eukaryotic cells, and we had a cyanobacteria and an aerobic bacteria enter another bacteria, stayed there, and started contributing to the functioning of the cell, and that's how eukaryotic cells came into being. All right. So there are a lot of evidences of evolution. That is fossils, homological structures, vestigial structures, embryology, biochemistry. Okay. Then cladograms. You need to know how to read cladograms, okay? So they'll give you different milestones. These dots are actually milestones, and beyond that, those organisms have that particular trait, okay? So over here, if they are saying like vascular tissue, then beyond that, all these uh, groups they have vascular tissues. If this is seeds, then beyond this all the groups will have seeds in them. So you need to know how to read it, and beyond, after each milestone the organisms have that particular trait. They also show evolutionary relationships. Okay, classification. Classification is was always done. Aristotle was the one who said, "Oh, if you move, you're an animal. If you don't move, you're a plant." So there were two broad categories: plants and animals. Okay, but then slowly with time, as scientists became more observant, they understood that that's not the case. You have a lot of plants. You have a lot of animals that don't move, like sponges. They are animals. They don't move. Um, they just filter water to get their food. Then came along Carlos Linnaeus. He developed binomial nomenclature, and he came up with different five kingdoms and now six kingdoms. Okay, uh, classification is also evolving, and he was the one who's initially in the past a name of an organism would be a long Latin or Greek description, and he said that no, it he came up with binomial nomenclature where it would just be two word names. Okay, bi means two, nomial means name, two word name for all the organisms. Like for humans it is homo sapiens. Homo is the genus and sapiens is the species. So this is your hierarchy. You need to know this. You can't change the order. It'll always be domain, kingdom, phylum, class, order, family, genus, species. And if you remember one of the tables that you made in class, you need to know that. It's a very useful tool that helps you on many questions. So the name is made up of two words. The first one is genus, the second one is species. So look at this one. It shows you only five kingdoms, but we have six. Okay, Monera was there in the past. Now we have Archaebacteria and Eubacteria. Archaebacteria is the ancient one that can live in very harsh climatic conditions, whether it's methane-laden environment or very hot or very cold or very low pH or very high pH. They're everywhere. So they are called Archaebacteria, and the modern day bacteria are called eubacteria. Okay, so they do not have a nucleus. They do have a cell wall that is made up of peptidoglycan. They are unicellular always, and they can either be autotrophic or heterotrophic. So if you look at that table, all of that is compiled over there. Okay, then your next one is eubacteria. They are similar, but they are the modern day bacteria, and they can survive and thrive everywhere. You need bacteria for your own survival. Your stomach or your gut has a lot of useful bacteria that helps in the digestion of your food. Protista, the mixed group. Any organism that did not fit into archaebacteria, eubacteria, fungi, plants, or animalia, they were put into Protista. Okay, it's like a mixed match. They are like eukaryotic. They have a nucleus. They do some have cell wall. Some are unicellular. Some are multicellular. Some are heterotrophic. Some are autotrophic. There's another one called Euglena. It's so unique in that it during daytime it acts as a plant and it has chloroplast, and during nighttime it becomes heterotrophic. It goes hunting like animals to find food. Then you have fungi or fungus. It was initially clubbed together with plants because they couldn't move. Then they learned that okay, they don't have chloroplast, they do not make their own food, they are not autotrophic, so they gave a special kingdom to it. So mushroom is we a lot of them are used as food for us. They are also decomposers. They do have a cell wall, but the cell wall is made up of chitin, chitin. Some people also say chitin. They are usually multicellular and they are heterotrophs. Plant kingdom is the only kingdom I would say that is all autotrophic. Okay, they have like and they have evolved. They have really evolved from the water onto land. They are eukaryotic. They have a nucleus and they have a cell wall made up of cellulose. Animal kingdom, they are multicellular. They are the only ones that do not have a cell wall. Okay, they all are multicellular and they are heterotrophic. This one is a dichotomous key. Di means two, cotomous. This is how initially in the past scientists would identify organisms, and they would like create a key to lead the scientists to the different organisms. You will not be asked to make one, but you should know how to read one. Okay. Now, types of plants, like I said, that plants have evolved. They were the first plants to live were in water. Then they started living near water. They did not have seeds. They would have spores, so they could like swim in water. They perform sexual reproduction. You know, they have male and female parts. So the sperm needs to swim to the egg for fertilization. So the initial plants were in water, then they started living close to water, and the biggest challenge for them to live on land was two main things. How will the sperm travel to the egg? Okay, and how will they prevent themselves from drying out? So they came up with like different types of adaptations, and that's how the modern day plants they live on land. Gymnosperms, they are unique. They don't have fruits. Their seeds are naked. Okay, that's gymno means naked, sperm means seed. So they have naked seeds. They are vascular. They have xylem. They have phloem. These are the vascular tissues, and they have their leaves are like needles and their seeds are in the cones. The cones are different. The female cone is big in size and you born singularly. Okay, they are single born. Whereas males, they are multiple small cones and they create a cloud of pollen. You know, it's like a cloud of pollen that just gets dispersed by wind and it goes and it travels to the female cone and pollinates it to form seeds. Angiosperms are the modern day plants. They are vascular, they have xylem and phloem. Xylem carries water from the roots to the different parts of the plant, and phloem, it takes sugar that is created by the leaves and distributes it to all the parts of the plant. Okay? So xylem and phloem together, they are called vascular tissues, a vascular bundle, and that is necessary for the plants to survive on land. They also have fruits, they flower, and they need to be pollinated. So plant adaptations, like I said, they roots, stem, and leaves. Roots, they absorb nutrients and water. They have a lot of root hair also that increases the surface area. Plant leaves, they perform photosynthesis. They have chloroplast. They have stomata on the underside that helps in the exchange of gases and loss of water called transpiration. And the stem, it helps in the movement of water and sugar. Okay. So if you see over here is pollination is happening. You did draw the diagram in class where we talked about the male and female parts of the flower. You have pistil, that is stigma, style, and ovary, that is the female part, and then you have anther and stamen, anther and filament, that is called stamen together, and that's the male part of the flower. Okay. Types of animals. It's very versatile, and uh, there are different types of phylum, Annelida, insecta, amphibian, and mammals. They all have different characteristics. Broadly, you need to know that uh, insects are said to be the most abundant and has a lot of diversity. Mammals, they are usually big. They have mammary glands to feed the young ones. They are said to be case strategists. They bear very few young ones during their lifespan, and they actually provide parental care to their offsprings. Whereas insects and worms and amphibians, they don't take care of the young ones. They produce in large numbers, and a lot of them they don't survive much. A few of them attain adulthood, and within a very long, they have a very short lifespan also, and they reproduce very early. They are said to be R strategists. Now, virus and bacteria. So let's talk about virus. A virus is not said to be living because of multiple reasons. Okay, it does have a DNA or RNA, but it can't reproduce on its own, nor can it metabolize. It needs a host. Okay, since it can't grow and develop on its own, it's not said to be living. So it has to have a host to grow and develop. It has a DNA and RNA. So if you see over here within this, it also has a protein called capsid, and either DNA or RNA. Okay, not both, either only one. And they can reproduce in two different ways. One is lytic cycle, the other one is lysogenic cycle. So when the new virus buds off and the cell bursts, releasing a lot of virus, that is lytic cycle. So here they have shown you the lytic cycle. You have a bacteriophage coming and sitting on the bacteria. It puts its DNA inside. The capsid doesn't go. It just stays outside. Once the DNA goes, it makes multiple parts of itself, and then it assembles itself. Once assembly has taken place and these viruses are ready to attack other fresh cells, it just lyses or it breaks, and it releases all these new viruses to go and attack other bacteria or find other hosts and go on that same destructive path. These viruses, they are unique. They have a protein that is host specific. The first virus to be studied was tobacco mosaic virus. That is a disease in tobacco plants, and they discovered that they were like host specific. The protein code that they have, the surface protein, they attach to specific shapes, and they do not like jump host or something. Okay. The lysogenic one is different. It stays in an organism for a very long time, so the DNA will enter, and it will like, let's look at this one. Yes. So this is the lysogenic one. So if you see, the DNA gets incorporated in the host, and it just multiplies and multiplies and multiplies and stays in the host for a very long time until something comes and triggers it to go into lytic cycle. Then it goes into that lysis or that destructive phase. So it could be anything in the environment, it could be stress, it could be chemicals, anything can trigger it to go into the lytic cycle and become virulent. Okay? Virulent is the word. So the difference between bacteria and virus, well, bacteria is living, we all know that, right? It has two big kingdoms in its name. So bacteria is can be beneficial to us too, like I said, that you need it, and plus it's a pathogen also. It can give us diseases, but for virus, it's not made up of cells. They're very small. They're much smaller than bacteria. You can't like treat them using antibiotics. Vaccination is the only way to fight viral attacks. Okay, it's a small dose of dead or weakened virus is put into your body produces antibodies and gets ready for future exposure to that virus. And when you're exposed, your body is ready to fight it, and you don't get that disease, or it's not very severe. Now, some diseases, like these are internal and external factors in certain diseases. If you see, malaria is actually transmitted by a mosquito. The mosquito does not give it to you. It is actually a protista that gives you malaria. It's called Plasmodium vivax. Okay. Then you have another one called PKU. It's a disease in which a person cannot break down phenylalanine, and it causes a lot of buildup, and it can cause mental retardation. Another one is lung cancer. Cancer is uncontrolled growth. If you remember the cell cycle, there are three checkpoints. So the checkpoints don't function, and the cell goes into an erratic division and forms a tumor. Now that tumor can go and breaks and goes to another part of the body and starts growing there. It's called metastasizing. And at that point, it can go and start growing anywhere. So, lung cancer can be triggered by smoking or by different chemicals. Diabetes, it is where your pancreas uh, do not make enough insulin to regulate the sugar in your blood, and this can make a person weak. It can make a person's kidney be damaged. So diabetes can is of two types. It's one is adult diabetes, and there's another one where you can get it at a very young age. Okay. Skin cancer is called melanoma, and this can happen when you are exposed to sunlight, and sometimes it can trigger your skin cells to start dividing. Melanin is the pigment that gives you the coloration in your skin, you know. So that is actually, it protects you. It protects you from UV radiation. It protects you from getting skin cancer. So it's good news to have more melanin in your skin. Now, immunity can is helped by B cells, T helper cells, and T killer cells, and these are all made by your white blood cells. Okay, so it says that B cells it makes antibodies, whereas T helper cells it helps coordinate the immune system, and T killer cells it kills the virus infected cells. Now, there are two types of immunity. One is passive immunity, where the passive one does not make memory cells or antibodies. It just antibodies are only transferred, and it doesn't provide you long term like protection or something. Um, a natural way would be like a mother breastfeeding in the initial days of childbirth. And then active immunity is where a person does make antibodies and there are memory cells, and that is what vaccination does for you. It provides long term immunity, and it protects you from the viral attacks or diseases. So animals, you should be knowing about the animal kingdom, their animal behavior. The main purpose of animal is to like survive and reproduce. So they need to protect themselves from predators. They need to find food, shelter, and then they need to reproduce so that they can further their progeny. So let's look at this. Innate behavior is something that is inborn, like hunting or suckling. Uh, all of these are like it's programmed in the DNA and it's there. It's like a reflex action. Some that animals do. Migration is another one, where birds know that they need to migrate and fly from point A to point B when the climatic conditions become harsh, or they go into hibernation so that they can overcome very cold climatic conditions. Estivation is the opposite of hibernation, where a lot of organisms in the desert and all they estivate. They are dormant during daytime and they don't move too much and they conserve energy. Okay. Learned behavior is where you train an organism. So you can train a rat to eat cheese or something. You can train a dog to jump over hoops. You can train your dog to come for food at a particular time. So imprinting is another one. So all of these are like learned behavior. It's like humans innate behavior is to speak speech, right? But what language you speak is learned. Speaking is innate, but the language is said to be learned. So the social behavior is communication. You know, bees they have a certain dancing style that they dance, and they signal other bees. They also create certain chemicals called pheromones, and they use it for like courtship, and they use it to attract mates. And then a lot of animals they are territorial. Like you can't have like friends between tigers or cats. They are territorial and they mark their territory. Now, this was ecology. It's we have talked about it in class where there's a biosphere. This biosphere is made up of different types of biomes. Biomes have different ecosystems, and ecosystems are made up of multiple different communities. And this communities is made up of by population, and population is made up of by the same species. So now, when we talk about ecology, it's about how organisms interact with the abiotic factors or with their environment. So we have food chain and food web. We have different trophic levels. There's a 10% rule also. Usually decomposers are not shown on the chain because they recycle nutrients and they could actually be in all the trophic levels, I think. So you have the first trophic level is producers. Producers can be just not just plants, but algae is also a producer. Uh, some bacteria they also producers can be photosynthetic or chemosynthetic, remember, where certain chemicals can be changed into organic molecules by certain bacteria, so they are chemosynthetic. Then these producers are consumed by primary consumers, then secondary consumers, then tertiary consumers, and then quaternary consumers. Usually you don't have more than four trophic levels because of the 10% rule where energy gets lost as we move up the trophic level. A lot of this energy is used for metabolic activities and is also lost as heat. So you could actually like create arrows here where it would be loss of heat from each of these trophic levels. Okay, so it would be loss of heat from each of these trophic levels. Here it says, okay. So this is the pyramid of energy. You have pyramid of biomass and pyramid of numbers also, but technically, but a lot of times when we talk about food chain um, or the energy pyramid in ecologies, in order to understand uh, the 10% rule or the law of thermodynamics. Symbiosis means living together. So when we talk about symbiosis, when two organisms are living together, it could be mutualism, where both of them get benefit. It's a relationship where both are benefited and they are interdependent on each other. Uh, and there are a lot of classic examples like lichens, the patches you see on tree barks or on rocks. They are they are very That's a symbiotic relationship between a fungi and an alga. The fungi breaks down the rocks and provides minerals and nutrients to the algae, and algae performs photosynthesis and provides carbohydrate to the fungi. Okay. Parasitism is where the parasite benefits it and the host is harmed. It becomes weak. They may not kill their host, but they certainly weaken them. These parasites can either be within their organism or outside. So when they are outside, they are called ectoparasite, ECTO, ectoparasite. And when they live within the organism, they are called endoparasites. So a lot of worms, like tapeworm, they are endoparasites. Okay, ticks and fleas and lice, they are ectoparasites. Did you know mistletoe is a parasite? They bore into the stem of their host plant and they suck nutrients from them. So a lot of plants are also parasitic. Commensalism is debatable. This is said to be a relationship where one gets benefited, the other one is neither benefit nor harm. But a lot of times we come across where uh, it's it becomes a mutualistic relationship. But there are a lot of examples where, like an orchid, it just perches on the tall trees in order to get sunlight. So that is a commensalic relationship where the orchid just gets perched and it grows. It doesn't harm the tree, but it gets sunlight, so it gets benefited. Okay. Predation and prey, usually you'll see that it goes hand in hand, and the curve will always move together. There's a direct correlationship between the two. When there are too many predators, then they eat a lot of food, so the numbers go down. And when then the prey is not there, so the predators they die of hunger, so their number comes down. So when the predators die, again the prey, the number goes up. When they get food, the predator's number goes up. So this is like a cycle that goes hand in hand for predator and prey. Then you have these two population growths. One is said to be a J curve, or also called exponential curve, where growth is unrestricted. Usually this type of curve is noticed in our strategists, and then there is when the resources become limited, it just crashes. Okay. Then the S curve is said to be the logistic curve, in which there a carrying capacity is reached. A carrying capacity is the maximum number of organisms an environment can support. The carrying capacity is never a straight line. It'll go up and down, up and down, up and down, up and down. It'll be within a range, okay? And it maintains it, uh, and that is said to be a logistic curve or a J curve. Human population has been seen to be a J curve, and there are a lot of reasons for it. Um, and it's very debatable whether humans have reached the carrying capacity or not. But we are leading to a lot of like resource stress, freshwater depletion, soil degradation, different types of pollution, air pollution that is leading to acid precipitation, or it is also leading to ozone depletion by the use of CFC, which has now been banned. It is also leading to eutrophication because we are growing a lot of crops and we are using fertilizers that is being carried into the water, and it is leading to algal bloom. That algal bloom blocks sunlight, and when it blocks sunlight, it kills the organisms that is in water, and it creates an oxygen sag where there is like a demand for oxygen, and a lot of organisms die because they don't get the oxygen. Okay. Then there's another one where it's called introduced species. It's also called invasive species. When they come in, what they do is that they don't have a natural predator. So since they don't have a natural predator, what happens is starts using the resources, and it outcompetes the native species. So the poor native species, they don't get like food, uh, and then slowly they are forced to reduce in number, and there's a loss of biodiversity. You all were given a project to do based on different global environmental issues caused by human population. So try to go and see your presentation, and that could help you in understanding this. So the next one is biogeochemical cycles. There are three main ones that we use in ninth grade: the hydrological cycle or the water cycle, the carbon cycle, and the nitrogen cycle. Okay, so we need these three, and they cycle between living and non-living. Earth is a closed system. Nothing is coming in except for sunlight. Okay, that's the only energy that comes in. Nothing leaves Earth. So all the material is just keeps cycling between living and non-living and gets transferred and transformed. So over here also carbon is within living things. So plants take up carbon dioxide, right? And then they also release carbon dioxide during cell respiration. A lot of carbon is also picked up by humans. So combustion, a burning of fossil fuels, releases carbon into the atmosphere. So greenhouse effect is by one of the issues caused by humans burning fossil fuels, and carbon dioxide is a greenhouse gas that holds on to heat, and that holding on to heat is making the earth warmer, and the temperatures are rising. It's not just carbon dioxide, actually methane also does the same thing. Water vapor also does the same thing. Okay. Then again, we need to be good stewards. We need to take care of Earth. We just have one Earth or one biosphere out of the 8 planets we have. Life is only on Earth. Water is limited. Fresh water is limited. We don't have too much water, fresh water. We do have lots of water, but fresh water is limited. So with this, we can recycle, we can reuse, we can refuse. We need to be careful in being a consumer and make smart choices um, in order to save Earth. Uh, So with this was the last slide, children, but I think there are more topics to be covered, like succession. Ecological succession is where life starts on bare rocks where nothing has ever existed before. So lichens and mosses are said to be pioneer species. They break down the rocks and lead to poor soil, and then different herbs start growing there. Those herbs, they die and they decompose, then they add to the soil. So organic material gets added to the soil. The soil becomes a bit better in quality and is able to support some shrubs, a bit bigger plants. So they also start living, and then they die and they decompose, they add to the soils, and then they get succeeded by trees. And then with the passage of time, that bare rock uh, converts into a thick, lush forest that is very biodiverse and very complex, with a lot of complex food web. And this whole journey from bare rocks to climax community takes hundreds and thousands of years, depending upon the climatic condition. It's not like 10-year or 20-year process, okay? And that is called primary succession. So again, this primary succession may be destroyed by something, by either natural forces like flood or mm, forest fire, um, or drought or something, or by humans. Humans lead to deforestation, right? But in this whole destruction, the soil is still there. Something that took hundreds and thousands of years to form still remains. So only the biota is destroyed. So again, life can start on the soil, and that would be called secondary succession. Okay, so I hope this helps. I tried my best to make it simple for you to understand, and there are a lot of videos that are available. You can watch those and take notes on what has been talked about in this video. And I wish you all the very best for your upcoming exam. Take your time to answer the questions, do not rush, and do your very best. Okay, thank you. Bye bye.